Let's begin. Heavenly Father, thank you for today. Thank you for giving us knowledge of this world and all that you've made and how it works, Father. And thank you for giving us wisdom about how we ought to live and what we need. Pray that today, as much as we learn about bacteria and archaea and viruses and immune systems and all these facts, Lord, that we would gain wisdom for how to treat our bodies, how to live in life, the things that we need, the things that we don't. Lord, knowledge that we gain just be very useful to us. Help us to learn it well. Help me to be clear. Help us to be wise in all these things for our good and for your glory. I pray this in Jesus' name. Well, today we're going to do prokaryotes, okay? Last class, what did we do? Viruses, which are not alive. They're not alive. We started things that are not alive. It's a contrast. It's a good place to be. It's also very important because they greatly affect things that are alive, right? In fact, we didn't just do viruses. We did viruses, viroids, and prions. Prions, think of the P. What are they made of? Proteins. Good. Viroids, what are they made of? Nucleic acid only. And a virus has both. Okay? That's good. All right. These things they're not alive, right? On their own, they cannot do all the characteristics of life. What can they do?
Grow.
Huh?
Grow.
They can't grow. They can kind of move. Not on their own. But they do reproduce, but not on their own. What else? In fact, let's talk about these things. So we've got grow, we've got reproduce. What else? Move. Use energy. What else? No. One more thing. Respond to a stimulus. It's very sketchy. Do they reproduce? Yes, but not on their own. I'm gonna go ahead and just circle that. Grow? They don't grow. This is pretty sketchy. Okay, not alive. But they have certain things that living things do have, right? You just named two of them. Two molecules, two classes of biomolecules, proteins and nucleic acids, right? And so. But they lack, and you guys said this in the video, they lack cells. There's nothing that's alive that doesn't have a cell. Nothing. Nothing, nothing, nothing. So now if we're going from things that are not alive to things that are alive, then everything we're going to talk about must have a cell, right? And there are two types of cells. They are prokaryotic and eukaryotic. Good. Pro and eu, these are really good, useful Greek prefixes. If you ever become a Bible scholar or a linguist, you're going to want to know these. Pro means, remember, before. Before. And eu means, y'all are here. Hey. You gotta fix this real quick. So, pro means before, eu means either true or good. Okay, either true or good. And a karyote or karya refers to the nucleus. Okay, so these have two parts to their name: pro and karyote. Pro means before. And this refers to the nucleus. Literally, if you just I want you to think about this. Do the pencils down for a second. This isn't facts that you pencil down. Okay. This is just one of those things you need to know here, because this is me helping you just as a student, not as your teacher. Okay. When you learn just some of these prefixes and things and roots, I realize you didn't sign up for biology thinking, well, I'm going to go learn Greek. But if you'll take just a little bit of time to think about these, oftentimes the name will tell you what's going on. Okay, if you just know pro means before, if all you knew was pro means before, and then later on you're going, okay, which one of these does not have a nucleus, a prokaryote or a eukaryote? I guarantee you, if you remember that pro means before, then when you see prokaryote, it's going to trigger, it's going to remind you, oh, that's before the nucleus. Prokaryotes don't have a nucleus. Okay? So don't just gloss over when I try to pound some of this stuff in. Don't just gloss over it. It's going to help you. And then what's really wild is suddenly when you're taking another class and some of these, and I'm not even talking in science necessarily, and you see these things and all of a sudden you know stuff that other people don't know because you're paying a little bit of attention in biology one day. So, okay, go ahead. We're back to it. Before the nucleus, right? These things are alive. They must grow, reproduce, move, use energy, respond to a stimuli. Okay? Two major groups within the prokaryotes. Okay? So let me do this. Life. All life. So everything that's alive is going to branch off of this, okay? You with me? I think we did something like this. All life is going to split into two groups. What are they? Help me. Prokaryotes and eukaryotes. This first classification is at a level we call the domain. Okay. You want to know that. You guys remember classification maybe from classes you took in high school or something like that? Kings, play cards, kingdom, phylum, class, order, family, genus, species. You all remember that? A little bit? This one is a level, they used to do it that way, kingdom, phylum, class, order, family, genus, species, and that was it. But as we've discovered more and more organisms and looked more and more at things, we've added a domain level, okay? Where we're just splitting up into right away into prokaryotes and eukaryotes. Do we have a nucleus? Do we not have a nucleus? The next level is the kingdom level. Kingdom level is where prokaryotes split off into two groups. One is archaea and one is bacteria. Can you name any of the other kingdoms, the ones under eukaryote? Yeah. You looking at notes if you have that. She looked at notes. All right, Protista. What else?
Fungi.
Fungi. What else?
Plants.
Plants and animals. Good. Very good. Okay. So these are the kingdom level for the eukaryotes. What's the difference between Protista and all the other eukaryotes? Between protists and all the other eukaryotes. Truly multicellular. Truly multicellular. Truly multicellular. Not truly multicellular. You may find them sometimes multicellular, but they don't have to be. Even the ones that are multicellular, you can split them apart. They don't have to exist that way. They can, it's beneficial, but it's not necessary. It is necessary for your cells to form into tissues. It's necessary for your tissues to form into organs. It's necessary for your organs to form into organ systems. It's necessary for your organ systems to form an organism. If you're not multicellular, you're not alive. We can't just be a one-celled human, right? Not past the zygote stage. Which we'll talk about later. So these are weird. These things are unicellular. The difference between these and this is?
What's that?
No! The difference between these and this is? Look at what I'm pointing at, right?
Nucleus.
The nucleus. Now, come back to me. I'm going to make you right. What's the difference between this and these? Multicellular. Okay, good. All right, no problem. So today we're dealing with these two guys. These are archae. They're also generally referred to as archaebacteria. Same thing. What's key is this Greek word, archae. You've heard of archae almost your whole life, at least as a root in other words. Can you name words that have archae in it?
Archaeology.
Archaeology. What do archaeologists study? Bones. What else? Fossils. What else?
The past.
The past. They study things that are ancient. They study. That's the core of this word. Archae are ancient. John, because I'm a pastor, so I'm going to go back to the Bible, right? So John chapter 1, verse 1. En archae ein ha logos kai ha logos en proston theon. In the beginning. En archae. In the beginning was the Word, and the Word was with God, and the Word was God. So beginning, ancient, old. That's the idea here. Okay? So we've got ancient bacteria, and we've got bacteria, and that doesn't really tell us anything. But it does separate them. So just know that that's the thing. We'll look at the differences between these two in a little bit. Okay? So far, so good? Okay. We've done a lot of Greek this morning. Close your eyes. Close your eyes. Everybody close your eyes. Pro. What does it mean? Before. You. Good or true. Archae. Ancient, beginning, old, something like that. Karya or karyote. Nucleus. Good. Open your eyes. That's good. Yeah. That if you really know that, that's going to help you to construct this and to know this when the time comes. Okay. All right. Now. Last time we looked at viruses. Virus is not alive. Virus is very important. They cause a lot of disease, a lot of trouble. These things are alive. These are very, very simple, very, very small. Everything over here is unicellular. These guys are not a problem to you. These guys can be. You will almost never interact with archaebacteria. Okay, let me just tell you that right now. These live in extreme environments. Okay. What do I mean by, what are some of the most extreme environments you can imagine on this planet? Radiation. What else?
Like Antarctica.
Okay. Freezing cold. What else? They don't live either of those two places. What else? Another extreme environment.
Extremely hot.
Extremely hot. Like how hot? What's the hottest thing on Earth?
Okay.
It's the hottest thing on Earth. Lava. Yeah. What's the difference between magma and lava? Fun fact.
One is solid and liquid.
Nah. Only difference between magma and lava is that magma's below the crust, and it's like it's rising up, right? Here's the crust. It's rising up. It's magma, 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 lava. As soon as it breaks the surface, we call it lava. It's the same stuff, okay? Crazy hot. Crazy hot. These guys look around magma. When magma reaches the surface, like in the ocean, and it comes up, and there are these gases and stuff that are released, especially lots of sulfur gases. These guys live there, where it's beyond boiling hot, and they can survive. And they use the chemicals that are in those gases. They use that as fuel, as energy to make sugars. So these live in extreme environments, especially extremely hot. Okay, for example, what we call thermal vents, or deep sea thermal vents. Okay, crazy hot. How do they survive? And by the way, well, here's another fun fact. How far in meters, I'll take it in feet also, how far can you go below the surface of the ocean before there's no light? Just do pitch black.
You know, like 8,000 feet?
It's not nearly that deep. It's far closer to the surface than you'd imagine. Because light just doesn't penetrate through that water. How far down do you think you can go? Where, like, seaweed couldn't grow below this depth. That's essentially what I'm asking. At what depth can you not grow seaweed anymore? You ready? 10 meters. 10 meters. So that's roughly 30, 31 feet, something like that. That's not that deep, is it? These things can live like thousands of feet deep. There's no sunlight. I think I talked to you about this last time, how as we develop technology, they're just like, what was that? Why are there shrimp? Why are there starfish? Why are there crabs? Why are there all these things? What are they eating? How are they alive? And that sure looks like a tiny volcano. How are they surviving this? Because archaea survive in extreme environments, and they go through chemosynthesis. This is more Greek time, right? You've got to know these two words. Chemo means, it is like it sounds, same in English as it is in Greek. What is it? Chemical. And synthesis, do you know what that word means? No, not. Synthesis. That's more of an application. The root word itself. No, that's kind of the same idea. That's to put, you're applying it. Yeah, photosynthesis, chemosynthesis. That's taking the root word and applying it. I'm trying to think of other words that have this root in it in English, and all of a sudden I know there's tons of them, but I can't think of a one right now. Syn means together. S-Y-N means together. Yeah, all of a sudden my brain is just not working. I can't think of a single English word with that in it. There are many, many, many. To synthesize, yeah. So chemosynthesis, it actually means chemical together. It means to put chemicals together. That's what it means in Greek. Now, how do we translate it? What does it actually mean for us? It means that you're using chemical energy to make sugars, make carbohydrates, make glucose. Plants use the energy of the sun to put carbon together to make glucose. Many bacteria, not all, but many bacteria use the sun to, as an energy source to put carbon together to make glucose. These things use chemicals to do that, the energy and chemicals to do that. You see them in deep sea vents. You see them in other places. You see them in hot springs. Has anybody ever been to Yellowstone National Park? You got to go in your life. Okay, this is amazing. There's in particular, this is where like Old Faithful is. You heard of Old Faithful? The geyser that erupts like every hour on an hour or something like that. Old Faithful's cool, but I mean you're just going to see water shoot up in the air. There is something there called Grand Prismatic Spring. And it's a natural hot spring, crazy hot water, right? You wouldn't want to go for a swim because it's crazy hot and toxic. But it's hotter near the bottom than it is near the top. I'm going to show you a picture of this because I got to show you some stuff anyway. So remind me and I'll show you a picture of Grand Prismatic Spring. It is beautiful. It looks like a flower or something, just like all these colors. And what it is you have archaea living in it. Different archaea living at different temperatures. Near the bottom is the hottest. So you got one group of archaea that can live there, one species. And then as you rise up, it's not as hot, right? And so then you've got another species that lives at that temperature, and then another species that lives, and then another, and so you just have these bands of color in this hot spring. And they're all archaea. And even as, because it's all hot springs around there, all the water is blazing hot and fed by chemicals and all this kind of stuff, archaea thrive. Even like the rivers and streams, you'll see just, you know, where they've just grown up over years, and there's just piles of them, okay? Awesomely cool. Very important in certain terms of ecology, because other organisms can eat them and survive and thrive where you couldn't otherwise. Fascinating because of chemical energy. Not that important for you. You're probably, unless you break the law and go scrape some out at Yellowstone or something like that, you're never going to touch one of these. You're going to have almost no contact. These don't cause you any disease. They're not of any economic importance. It's not like you can go harvest the shrimp that grow around deep sea vents. That would be crazy expensive and stupid. Fascinating to you in your life, not that important. These are. Okay? Did I mention these don't cause you any diseases? Like, make a note of that. No, these don't cause any diseases to you. They are not a problem to you at all. You have no archaea diseases. Can you name the disease of bacteria, though? No, that's viral. What else? Throw stuff out. Let's see what sticks. Tonsillitis is likely to be caused by bacteria. Likely. What other diseases in that area? What does everybody get in their throat? Strep throat. Streptococcus. What do you get in your lungs? That's a human disease. But pneumonia. Streptococcus pneumonia. Diseases on your skin, staph infections. Diseases from eating spoiled food, botulism. These are diseases that are caused by bacteria. Bacteria can cause lots of problems. But on the other hand, you can't live without them. You must have bacteria. You must have them particularly in your gut. Right? There's a whole industry about probiotics. What does that word mean? Put it together. Pro, before, biotic, bio, life. Before life. These are things that you need to live. Now, when they talk about this, they're saying, hey, you need these bacteria. You need they're saying, just the very name probiotic is you need bacteria to live. You can't function without these things. You need them in your gut. You do not produce vitamin K on your own. There's certain B vitamins you don't produce on your own, but bacteria in your gut produce them for you. So you need them and they need you. They need what you eat and you need what they secrete. So we call that a symbiosis. Let's put that word out there. By the way, this prefix and this prefix have the same root. So what does symbiosis mean? What does that mean? If it has the same root as sin,
Be together.
together. Together, and what is bio? Life or living, so together living. Symbiosis is together living. Two things living together. You have a wonderful symbiotic relationship with many bacteria. You have a rough symbiotic relationship with other bacteria. Some things are parasites to you, some things are highly beneficial to you. I'm not going to test you over this right now, but it's one of these things that's going to come up a lot. It's a great example, so I'm going to talk about it. You get strep throat. What do? Panic. You go to the doctor, he prescribes antibiotics. Let's talk about that. Anything I'm writing down, you need to write down, okay? Anti. Yeah, that's how we use it. That's actually not right, by the way. Like in the Bible, when it says Antichrist, it doesn't mean the one who's against Christ. It means the one who puts himself in place of Christ. The one who puts himself up as a false Messiah, a false Savior. But for this, against works. These are things that work against life. Okay? So you take antibiotics to kill bacteria, right? Okay. We're going to talk about this. Is going to come up several times today. Antibiotics function to kill bacteria. You need them because you're not doing well. You're getting your butt kicked by your strep throat, by your pneumonia, by your whatever. These things, if you're healthy, your body can probably fight these bacteria off by yourself. If you're really young, or really old, or really sick, you may not. Very few people your age will ever die of pneumonia. Babies, ill people with cancer or something else, elderly people die of pneumonia all the time. Your bodies are in the prime of all their capabilities to fight off anything and everything. And so you'll be sick probably for a long time, but eventually you'll win the war. If you're a baby, an old person, somebody who's immunocompromised, you need help. And so we take antibiotics, which do you know where antibiotics come from? Is it like penicillin or something like that? Fungi. They come from molds in particular. Okay. It's crazy. I'm going to take a side. Can I take a side? I'm doing it. You're here. What are you going to do? All right. So, grow something in a petri dish. Okay. Here's my petri dish. Okay. I got Streptococcus pneumonia growing. Okay. Here it is. Here's my Streptococcus. Maybe I got two of them growing. Two little colonies. Two little cultures growing. Right. Over time, they're just going to grow. Okay? And then I treat this with a little bit of mold. So I put some mold here. Okay? These two, this is literally what happened. This is how they found this. A scientist had, I don't remember which bacteria he had on his petri dish, but it got infected with a little piece of mold. And instead of just throwing the petri dish out, he wanted to see what would happen when they hit. And so he watched it. Well, what happened was crazy. As the fungus grew, and as the bacteria grew, we're wherever they touched, the bacteria died. Wherever they touched, the bacteria died. That was amazing. No one had ever seen that before. Well, what is it about this mold that's causing this bacteria to die? And how do we get that? Well, what he was studying was penicillin. Penicillium. It's a mold. It's a common mold. In fact, the penicillium that was used to make penicillin back in World War II, they would harvest it from cantaloupes at the grocery store. It was just growing wild on cantaloupes. And so you go scrape a cantaloupe with like a Q-tip or something like that, and you put it on your petri dish, and you culture it, and it grows. And you find that it produces some kind of a chemical that kills bacteria. And we call that chemical that kills the bacteria an antibiotic. In this case, the antibiotic we call penicillin, right? There's also amoxicillin and all kinds of stuff. And that goes like, here. Bottom line is, antibiotics stop bacteria from growing. Crazy cool. Now, taking all these disparate parts from all over, I need to try to put them together for you, okay? All this has to do with bacteria, but we need to make the parts fit together. So, we said that you have some bacteria that live in you and on you, and it's a symbiotic relationship. It's really great. In your eyeball, you have bacteria and you must have them. In your gut, you have bacteria and you must have them. On your skin in certain places, you have bacteria and you must have them. When you take antibiotics, it kills bacteria. So you got strep throat. Which bacteria did you want to kill? You want to kill the strep throat. Which bacteria did you kill? You killed the strep throat. What else did you kill? All of them in your throat, and where else? All right, let me give you this. Antibiotics are like a shotgun. What does a shotgun do? It just sprays out indiscriminately, right? There's something over there. One of these ought to hit it, right? But if I shoot, no offense, but if I shoot a shotgun at you from here, who else am I hitting? You and you and you and maybe you. I mean, I'm hitting everybody. I know you can't talk about shooting guns in college, right? But you get the idea. Whereas a rifle is super specific and targeted. Antibiotics are not super specific and targeted. They are very general. I don't only hit the bacteria that I want to kill. I kill all kinds of bacteria. I kill the ones causing my strep throat. I kill the ones in my eyeball. I kill the ones in my gut. If you just take antibiotic after antibiotic after antibiotic, you're going to destroy the ones in your gut. And now you're going to start to develop leaky gut. You're going to start to develop you're going to be lacking key nutrients. You're going to have digestive problems. You need probiotics. You've got to do something to get those bacteria that are beneficial, symbiotic, mutualistic with you that you killed. You've got to do something to get them back in you. And so probiotic is the answer. Ladies, this is very real life for you, okay? Gentlemen too, but more ladies. Remember, you have bacteria living everywhere. And a whole lot of it is highly beneficial. I'm just sitting here going, where is this other cap? I got marker cap sitting there. You got bacteria living everywhere. Most of it is highly beneficial. You take an antibiotic because you had strep throat and you kill bacteria all over the place. And now a week later you have a yeast infection. My wife just had to take antibiotics 2 courses in a row. She had 2 different illnesses back to back. Her doctor prescribed her something for a yeast infection in case it came on. Why? Well, what is a yeast infection? Yeast is a fungus that also lives all over your body. Gentlemen, you can get a yeast infection also. Athlete's foot, that's fungus. Ringworm, that's fungus. most of the time, these funguses that are in you, around you, on you, they're there, they're not causing you a problem because the bacteria are kicking their butts. They're out competing them for resources, for space, they're killing them, whatever. But now you kill the bacteria and there's no more competition for fungus, and it grows. And it becomes a yeast infection, it becomes athlete's foot, it becomes ringworm, because you change the landscape of your body. Not only are you a living organism, you're an environment. Just as much as the earth is an environment for living things, you are an environment for living things. You got so many, I think I told you this last time, you've got trillions and trillions of cells. But in terms of number of cells, not volume, not mass, but number, it's like 10% of human. You go killing all the bacteria, the fungus takes off, right? Did that make sense? I want you right now, just real quickly, write for yourself, hey, when I take antibiotics, here's why I need to be careful of a yeast infection, okay? Male, female, whoever you are, write that down. This is very real, practical life. When I take antibiotics, I need to be careful of a yeast infection because. Now explain, because antibiotics kill all the bacteria. And now there's no more competition for the fungus, and it. That wasn't too much. Now I want you to write one more thing. When I take antibiotics, I need to think about a probiotic because. When I take antibiotics, I need to think about a probiotic because. There we go. Okay. By the way, this is a picture of Grand Automatic Spring. You can that steam rising above it. You can tell it's crazy hot. This is like a boardwalk behind it. You can see the people. So if the people are that small, think about how big this thing is. And notice the color. You've got this deep, deep blue that's coming from copper that's in it. So these archaea that are growing there are just they're expressing that copper color. And then you've got the green, and then you've got the yellow, and you've got the orange, and deeper, and so this, the green and the blue are more based on the copper color than they are the actual archaea. This is probably too hot for archaea to live deep inside. inside. But when you get out to these rings, notice how the green starts to fade into some yellows, and bright yellows, and oranges, and dark. This is the archaea, right? So it is just amazing. And these are using chemicals that are there in the water as a food source. They do not do photosynthesis at all. Let me show you one more thing with this. See if I can just pull up like a little video or something like this.
The earth cracks open. Fluid and minerals spew from the seafloor. Water shimmers. Life abounds. We're looking at hydrothermal vents, originally discovered near the Galapagos Rift in 1977. Basically, a hydrothermal vent is a hot spring produced by underwater volcanoes or tectonic activity.
Go on. I'm gonna play.
Cool about it is the abundance and assortment of life that exists there.
That's octopus.
It's not life like we're used to up here on the surface. It's adapted to the dark conditions of the deep ocean. Organisms that live around hydrothermal vents don't rely on sunlight and photosynthesis. Instead, bacteria and archaea use a process called chemosynthesis to convert minerals and other chemicals in the water into energy. This bacterium is the base of the vent community food web and supports hundreds of species of animals. What kinds of animals? Well, scientists on the NOAA ship Okeanos Explorer went to the Rift in 2011. And here's just some of what they saw. Riftia tube worms, also called giant tube worms, which can grow to their almost 5 feet long in less than 2 years. Bathymodilus deep sea mussels, which are often the first creatures to colonize the vent, and are able to survive for a short time after the vent isn't active. Serpulid, or feather duster worms, and Tevnia tube worms, which are food of choice for vent crabs, the top predator of the vent community. But it's not all animals down there. Researchers also saw white flocculent material erupting from the vents like a snowblower, an indicator of microbial life beneath the seafloor. And of course, there were fresh lava flows, which means the seafloor is continuing to change. And that change means there's always more to learn. So join us as we continue to explore the...
Alright, that's enough. Anyway, that's kind of cool, right? Again, I showed you that because I said that I would. But we are on to bacteria. Okay. What do you need to know about bacteria? You need to know they have no nucleus, right? You need to know they're unicellular. Those things are just given, right? They're unicellular. You know that they use energy. How do they get their energy? Well, most of them not. We're not talking chemosynthesis. Some of them are what we call photoautotrophs. And others are what we call heterotrophs. So we're back to our Greek. This is biology, and these are just words you have to know. Troph, we see it in both, and it has something to do with energy, we know, because that's what we're talking about here. Troph means, taught you this on first day, might not remember, and that's okay. Anybody know what that? Pardon? Okay. It has to do with feeding. feeding. Heterotroph, autotroph, both have to do with feeding. Hetero and auto, these are, for purposes of this, antonyms. Auto, like autobiography, autodidactic. What does that mean? Auto. Who writes an autobiography? You do, right? It means self, right? So, so an autotroph is self-feeding. Heterosexual. What does that mean? Not two. What does the prefix homo mean? Homogeneous, homosexual, homozygous, same. Okay? And we contrast those to say homosexual or heterosexual. So same sex or heterosexual means other sex. Good. Okay? So heterotrophs are other feeding. Autotrophs are self-feeding. These guys are photoautotrophs. What does photo mean? Light. Good. Light, self-feeding. What do they do? Photosynthesis. Not all bacteria do photosynthesis, but some of them do. And not all bacteria eat other things, but some of them do. When you say heterotroph, all we mean is it eats. And some of them can do a combination of these two. So if I get down here, my archae, and I say chemoautotroph, means it's chemical self-feeding, right? Chemosynthesis, right? So that's what these would be. You could probably figure that out without me telling you, because you're learning Greek. You didn't even want to. All right. There's nothing to do but to learn this. But when you do, oh, it's going to help you. Scientists are not that clever. They're like, well, what does it do? Okay, let's just name it what it does. German's like that, by the way. The German language, they just, whatever something is, they're like, well, what does it do? And then they name it that, which is why their words sound crazy when you hear it. But when they hear it, like, the word just describes what it does. And you're like, oh, okay, good. We don't do that in English. We give it crazy names with anything, right? Why is it called a car? Yeah, what does a car mean? I don't know what a car means, but I know what an automobile means. What does auto mean? Self. And mobile? To move. It moves itself. A bicycle. What does bi mean? To. And cycle? To go around. So it goes around on two wheels, or has two wheels that go around. So when we name it in Greek, like all of a sudden you know what those words mean, you're like, oh, okay, that's cool. And we just throw the word car on it. What's a tricycle? It has three wheels, but if I call it a trike and you don't know what that comes from, you're like, I don't know what a trike is. That's weird. Anyway, there you go. So bacteria, unicellular, no nucleus. You have two types, photoautotrophs, you have heterotrophs, okay? These things eat, including many of them, you, okay? Real quick, I want to go back because I want to cover all of this. We talked about types of life, right? Or characteristics of life. We said it has to use energy. Well, that's what we just talked about here, right? Okay. What else do we say? It has to be able to adapt or respond to stimulus. What else do we say it has to be able to grow? What else do it have to be able to do? Move? Reproduce. Okay, we better talk about all these things. Okay. Let's talk about movement. Okay. Some of them don't move much. Not much. Is that where I want to go right now? Bear with me. I think we better talk about cell wall first. All bacteria have a cell wall. All bacteria have a cell wall. All bacterial cell walls have some level of a substance called peptidoglycan. This is very important for you to understand. Peptido, if you haven't had biology in a million years, you're not going to recognize the prefix peptide. If you have, then this is signaling you right away, hopefully, and you're going, hey, that stands for protein. Glycan, that sounds like other things, maybe like glycolysis, maybe like glucose, and I know that that is a sugar, and so you're thinking, okay, it's a protein sugar. All right, well, whatever that means. Peptidoglycan is a substance that's made up of a protein and a sugar that have been bonded together, and it functions as a building block. Think of it like a Lego. Their cell walls are built out of stuff. All cell walls of all bacteria have some peptidoglycan. Some. Some have very little, less than 10%. Some have all peptidoglycan. I keep throwing this word around, it really does matter, okay? It's a Lego. It's a Lego which antibiotics can destroy. Which is really important. So when we talk over here, well, how did penicillin destroy this? How did they kill it? It released an antibiotic that destroyed, or it inhibited the production of peptidoglycan. So if somebody wants to get into your house and rob you, but you've got, the walls barred, and you've got everything else, and they don't have anything but their fist, they're probably not getting in, right? But if they come in with a wrecking ball, they knock down the wall, and then they just walk right in, right? Or they knock down the wall and people come fleeing out because they're scared or whatever. Think about a water balloon. The water inside of a water balloon, generally speaking, is safe. Yes? Until the balloon pops, and then what happens? All the water comes spewing out. Same thing with the bacteria. The bacteria is very safe inside of its cell wall unless something happens to its cell wall. Once that's punctured, now the stuff inside is very, very vulnerable and can just go spilling out. Okay? So antibiotics function to break apart, or to prevent the cross-polymerization of peptidoglycan. It's like tearing down the walls of a castle. Okay? You familiar with a trebuchet? Oh my goodness. Do you even lift? It's the greatest. Okay, so here's a trebuchet. I guarantee you've seen it. If you've seen Lord of the Rings, or you've seen any like medieval knight type stuff, you've seen a trebuchet. Trebuchet has nothing to do with biology right now. This is just, you need to know this because you're a human. Because you'll have kids, and your kids should know what a trebuchet is. So you take a trebuchet, it's got like a locking mechanism, okay? So it can't move. It's got a lever arm here. And it's got a bucket, and you put a bunch of weight in that bucket. Now what does the weight want to do? It wants to fall. When it does, what does the lever want to do? It wants to move. The lever is attached to like a sling, and at the end of the sling is a big old stone, or if you're doing this in a high school robotics class or something like that, which I taught, it's probably just a tennis ball because you're not trying to kill anybody or get fired. So, but you set this up, and if you do it right, then when you release this, when you pull the pin, this thing goes falling with such force, and And this thing goes, and this acts to even extend the lever arm beyond what you thought you could. And this ball just goes, and they would shoot these at castles. You didn't need to have a huge trebuchet, you didn't need to have a lot of mass, because if you could shoot that projectile at enough force, you would just tear apart the castle walls. Now, you might think I wasted 3 minutes of your life, but all that was so that you could understand what is happening with an antibiotic. It is like hurling a stone at a castle wall, but it only works on peptidoglycan. All bacteria have peptidoglycan, but some of them have like almost all peptidoglycan in their cell walls. Some of them have like 10% or less. So not all bacteria can be destroyed by an antibiotic. Right? That's really, really important. So, cell walls, all made up of peptidoglycan. There are two major classes here. Gram-positive and gram-negative. What this refers to is what's called a gram staining process. So you take a bacteria, you take a smear of it, and you apply this dye onto it. And if it's composed of peptidoglycan primarily, that stain is going to stick, and the cell walls are going to turn purple, and you'll be able to see them very easily under a microscope. If it has very little peptidoglycan in it, then that stain's not going to stick, and you're not going to see it very well. It's not going to turn purple under the microscope. Okay, so gram stain is literally a stain that's going to stick to the cell wall if the cell wall has peptidoglycan. If you have some kind of an infection, there's bacteria that's evident, probably the first thing they're going to do is take your bacteria, they're going to smear it, take a little sample, smear it on a slide, stain it with gram stain, look at it under a microscope, because they want to know right away, are we dealing with something gram-positive or are we dealing with something gram-negative? The consequences are huge. Which one do you want to be infected with, and why? You want a gram-positive because it's made up of peptidoglycan. Why do you want that? Because your antibiotics will work on it. Like a trebuchet hitting a castle wall. If it's gram-negative, that's not going to do you any good at all. They're going to have to fight it with something else, some other kind of a chemical, and they might not. Let's talk about antibiotics one more time. We've talked about them a good bit. Number one, where do they come from? Fungi. Right? Think about that culture. That's how they were discovered. What do they target? Bacteria, which kind in particular? Gram-positive. Why do they work on gram-positive? Because they function like this to destroy peptidoglycan, right? Why don't antibiotics work on viruses? They don't have any peptidoglycan. There's nothing to destroy. Doesn't work at all. Okay. So if you get common cold, which is a virus, taking an antibiotic is not going to do you any good. You get the common cold, and then a week later, suddenly you have pneumonia, which is a bacterial infection. How'd you go from viral to bacterial? Just think of that. This happens all the time. Happens to me all the time. How do you go from a virus to a bacteria? Let me back up and make you think of something we talked about earlier. Who dies from a bacterial infection? Three groups. The really young, the really old, and the diseased, the sick. Why do I go from a virus to a bacteria? Because my immune system was compromised. I was so busy fighting off the virus that I got weak, and it gave room for the bacteria to take hold and grow. It's very, very common. Very, very common. And now take your antibiotics, right? Okay. All right. Where are we at on time? 36 minutes. I'm going to erase this side of things. What does auto mean? Other. What does troph mean? Feeding. What does chemo mean? Chemical. What does auto mean? Self. What does you mean? True or good. What does pro mean? Before. Good. What does troph mean? Feeding. Good. What does sim mean? Or sin together. Biotic. What does it mean? Life. Good. Anti for our purposes today. Against. Biblically. And everywhere else in Greek. In place of. Yeah. I would not throw that on a test for you, by the way. That would not be fair. But, in fact, I'm not going to test you on any of this vocabulary. You're just going to be dealing with the vocabulary. Okay. Moving forward. Prokaryotic cell. Every cell must have a... Nope. We're dealing with prokaryotes, no nucleus. They must have a what? Cell. No. You do not have a cell wall.
Membrane.
It must have a membrane. Good. Okay. All right. Here's my membrane, right? All. Say all. All prokaryotes have a cell wall. All of them. The cell wall is this incredible defense mechanism, this incredible structural mechanism. Provides, think about a plant, right? A plant without a cell wall, a tree without cell walls, it's just going to fall over, right? Provides structure, provides support. Is the cell wall going to be inside or outside of the cell membrane? What do you think? Outside. Okay. Cell wall. All right. These guys get a third layer. Capsule. So I've got a membrane, I've got a capsule, I've got a cell wall. What's inside the membrane? What's the liquid inside the membrane called? The only one in here who can do it for me.
Cytoplasm?
Yeah! Okay. So this liquid stuff inside is the cytoplasm. Okay? It's very liquid. Everything in here has got to be able to move. These things have got to be able to move and function and grow and all that kind of stuff. It can't do that if it's just rigid, right? So it's not rigid. It's liquid. No, this is rigid. It's good and it's bad, right? It gives us strength and protection, but it also limits us. You can't get that big. You can only get oh so big, and you can only move oh so much or oh so fast. Or you've got to overcome this, right? But inside we've got cytoplasm, right? And if you're going to live and you're going to grow and you're going to make proteins, you do all this stuff, what must you have? Come on. What must you have if you're going to make proteins? One blank equals one protein, central dogma. Central dogma says one gene produces one protein, and genes are found on your DNA, nucleic acid. Okay? So these guys have nucleic acid that's in the form of a ring. Well, let's do this. I'm going to draw some DNA, okay? Here's some DNA. I'm also going to draw this DNA. All the orange is DNA, okay? This is called this area is called the nucleoid. Not nucleus, nucleoid. Where's your DNA at? It's, well, I don't have a nucleus, we call it a nucleoid. Wherever your DNA is at. And we've got the bulk of the DNA, but then we've also got this weird little thing that we call plasmid. You're going to want to know that. That's important for why bacteria are what they are and do what they do, how they function. So nucleoid is my DNA. I've got my DNA hanging out there. I've also got this particular little ring called a plasmid. We've got structures called pili. Pili are like little tubes. I'm just going to draw a few, but these are all over, okay? Pili are important. They can help it to stick to stuff. If you're a bacteria and you're living in the gut of a human host, you know, the stuff's moving through that gut, right? Just constantly. Food's moving through, and your body's epistaxis is pushing stuff through, and you know, not to be crass, but like you poop. And every time you go to the bathroom, you're pooping out a lot of bacteria. But not all the bacteria. That bacteria wants to hang on for dear life. It's got to be able to stick to your cell surfaces. Pili are how it sticks. There's bacteria right now on your hand. There's bacteria on the surface of this desk. And if you go, it does not just blow away. It's not just sitting there like dust. It's sticking like glue because of pili. Okay? And by the way, even though what I'm about to tell you would sound like it's going to contradict what I'm going to tell you right now, still no, bacteria do not have sex. They do not have sex. They do not have, they don't go through meiosis and produce gametes, and I'm going to give you half of my genome and you give me half your genome, and we'll put them together and we'll make a little baby and it'll have recombinant DNA. They don't do that. They're asexual. You better know that. Asexual. This is more Greek. What does A mean in Greek? Not. It means not. These are not sexual. They don't do sexual reproduction. They just do reproduction. You ready? I'm going to show you. You're going to do it with me. Everything down for 1 minute. Hold up a little bacteria. Hold it up. Come on. Nice and high. This is asexual reproduction. You ready? Now this is your cell membrane. You can't just pop your cell membrane because what happens? It's like a balloon. Everything just oozes out. Ready? So let's do asexual reproduction. Ready? Go. One bacteria just became two. Listen. Ready? One more time. This is a bacteria reproducing. Know this. None of this. You with me? None of this. Just this. Okay. All right. Asexual reproduction. And yet, now I'm going to sound like I'm going to contradict this, they have something they call sex pili. But it's not sex. Okay.
Are you baking, sir, or are you fine?
I am crazy hot.
Okay. Well, I just went into the restaurant, like, oh my gosh, I'm suffocating. I'll see if I can fix that for you.
Okay, thank you. So, sex pili. You understand what happens in sex? When we say sex, what we mean is like, I'll give half my DNA, you give half your DNA, or maybe we'll have half of both of our DNA, it'll have a whole amount of DNA, it'll be genetically distinct from each of us. Right? And that's good. That's why you look like your mom and you look like your dad, but you're not a clone of your mom or a clone of your dad. That's why your brothers and sisters look like you, but they're not just like you unless you happen to have an identical twin. And no, by the way, boys and girls, like I have a set of twins, and it's just amazing how often you get asked, are they identical? One's a boy, one's a girl. You can't have identical boy-girl twins. So think about that for a minute. Yeah. So, sex pili. What happens with a sex pili is this. I'm going to draw another drawing for you, okay? So I got two bacteria and their sex pili come and they link. All right? And now I've got my nucleoid. I've also got this little plasmid ring. And we may exchange plasmid rings, or I may give you a plasmid ring and you didn't have one before. It's called sex in some way because it does involve a little bit of exchange of genetic material, but not all of it, and not half of it, that's for sure. And we're not making a baby out of this. We are exchanging a plasmid ring. Or I'm giving you one and you didn't have one before. This matters. You can have a whole petri dish full of bacteria. Here's the bacteria, it's just growing. And I can treat that with an antibiotic, and you know what's going to happen? It's all going to die, right? It's all going to turn black, it's all going to die, boom. Now, by that I can infer that this thing had a lot of peptidoglycan, it was probably gram-positive, and the antibiotic destroyed the cross-polymerization, all that kind of stuff. Have you ever heard of antibiotic-resistant bacteria? Yeah. You ever heard of MRSA? Methicillin-resistant Staphylococcus aureus. MRSA, M-R-S-A. M-R-S-A. MRSA is MRSA because MRSA, unlike regular Staphylococcus aureus, staph, like staph infection, unlike regular Staphylococcus aureus, methicillin-resistant Staphylococcus aureus has a plasmid that has a gene on it that changes the structure of the peptidoglycan so that antibiotics don't work on it anymore. And based on what you just learned, that means that if I have one Staphylococcus, or methicillin-resistant Staphylococcus aureus, one, and I introduce it to a colony, it can transfer its plasmid to the whole colony. And all of a sudden they can all be antibiotic resistant. So they test this. You have maybe 2 colonies on a petri dish. And this one over here, we introduce plasmid ring. And by the way, I don't have to put it in the bacteria, I can just put it in the environment. The bacteria can actually absorb the plasmid ring. It can bring it into itself. And it can pass it on to all the other bacteria. And then I come and I treat this whole thing with methicillin, and all of a sudden this whole group is antibiotic resistant, and this whole group dies. And that's because they pass it on through their sex villi. Isn't that a little wild?
Terrifying, though.
Pardon?
Terrifying too.
It is. What do I want you to know? What time is it?
10:50.
What time?
10:50.
Oh, goodness, okay. We'll pick up here next time. Okay. God bless y'all. Have a great day.